It's one of the 3 lectures regarding RNA sequencing experiment, and we will start from a review of what we have talked about in the last 3 lectures. Basically, those are the DNA sequencing analysis. And we talk about from the raw data, we need to do the quality control and quality filtering for the, all right, I'm going to turn that off. Otherwise, you will see every single of my email. I don't want to do that. Discard changes. All right. Okay. So we start from raw data, which is usually it's very big data sets, and then we need to do the quality control and the basic quality recalibration. Remember that every single platform, regardless of how honest they are still biased. So if they give you, they say the quality score is 30, that means the p-value is 0.001. That doesn't mean it's correct. And you should do certain level of recalibration because that will certainly affect the quality of later analysis. And next, you will do the alignment part, which we also talked extensively in the second lecture, and which we'll briefly touch base a little bit today as well. And we mentioned the importance of the refined alignment. And keep in mind that the initial alignment, that most of those short-read aligners, they are not doing very well in the insertion-deletion regions. So sometimes they really give you the false positive or false negative alignment there. And by doing refined alignment, which instead of maximizing the alignment for each individual reads, rather it's maximizing the alignment for the entire region, and then you can correct those very well, and a lot of false positive variations will be removed by that stage. And after you think your alignment is pretty comfortable, you can further do the variation call. And there's multiple ways to do that. And remember, if it's a diploid genome, which means for human, for example, it's diploid genome, and we have 2 chromosomes, one from mom, one from dad. And if they are reasonably balanced design, half-half, and then we can use just SEM tools to do that. It's pretty good. But if the design is not balanced, if we are doing cancer somatic DNA, which means only a small portion of the cells has that mutation, or if we are doing pooled DNA sequencing, which means only a small percentage of people in that pool has that variation, or if we are doing RNA sequencing, which one of the alleles is not as much expressed and not as much as the other one, and that is still diploid genome, but it's not going to be balanced design anymore. So there's multiple ways to do that, and we reviewed that a little bit, but that is still very active research area to go through. And after doing the variation calling, and we talk about the importance of variation quality recalibration, because many of the variation that has been discovered are later proved to be false positive because of the technical variations. For example, strand bias, that is one of the parameters that we can tune into. If you see the variation all happens in one strand rather than both of them, and if this is DNA sequencing, you may have a higher likelihood that it's a false alignment and there's some kind of artifacts there. So doing the quality recalibration is a very critical step, and I'm happy to see that one of the groups picked the project and further discover this part, and that is very nice. So after we do quality recalibration, and then we need to, let's say we identify the 30,000 variations in the exon sequencing experiment, but which one is potentially functionally important? So there will be, there's a lot of other statistical approaches that before this step, even though once we got hundreds of variation candidates, and we need to predict which one is potentially functional. And we talk about, that was the last lecture after Tatiana's part, and we talk about non-synonymous variations, which are the variations that potentially will change amino acid sequences and therefore change the protein structures and protein functions. And we talk about it intensively on the synonymous variations, which most people just discarded. And we talk about multiple molecular mechanisms that synonymous variations can be useful as well, and introduce some of the informatics tools that we can use synonymous variation to do the prioritization. And in the end of the last lecture, we briefly touched a little bit on the rare allele association studies, and those are still very active research area. But at this stage, I think we need to keep an eye on it, and there's multiple tools already being developed. Okay? So any question regarding the DNA sequencing part? I'll take that as a perfect. Okay? All right. So today, we will talk about RNA sequencing. So this is the central dogma of molecular biology, from the DNA to RNA, RNA to protein, transcription, translation, right? And the DNA sequencing part is apparently targeting this part, which is sequencing the RNA molecules. So we'll have 3 lectures on this. And the first lecture, which is today, we will talk about the overview of the RNA sequencing experiment. And I will introduce a couple of experimental considerations before you do the experiment. So this is not analysis part, just a very basic biological stuff. And I'm going to introduce a couple of published examples and to see how the RNA sequencing can change our view of transcriptome or in general biology. And I want to talk about, from the analysis part, a little bit on the alignment, because RNA sequencing, they have intronic regions and exon-exon boundaries, so their alignment can be a little bit tricky. There's a special strategies people have developed just fit the RNA sequencing part. And then I'm going to talk about the gene expression analysis and differential gene expression analysis. And this will be the topic today. For the next topic, next lecture, and the first half will be a guest lecture from Milan Rodvich, and who was a graduate student in genetics, but now faculty member in Department of Surgery. He's going to introduce using RNA sequencing data to study tumor, which is he's a very good speaker, and I really look forward to his lecture. And after that, I'm going to summarize the alternative splicing part, and this can be pretty difficult, and also the gene fusion part. So this is the next lecture. And the third one, I'm going to talk about allele-specific expression, which include the transcription variation identification, and some of the statistical approaches that's been developed in allele-specific expression studies. And I will talk about RNA editing, which is some kind of area I think is important, but I don't have any data because it requires very deep sequencing. And I will close that with non-coding RNAs, including the long non-coding RNA analysis and small RNA sequencing analysis. So this is at least my plan. I don't have any of these lectures prepared yet. This is my plan. Okay. All right, let's start from the overview of the RNA sequencing experiment. Okay. And when we think about the RNA expression, RNA sequencing, the first thing that we will jump into our head is we want to use this to study gene expression, right? And for example, there's 2 genes here, and there's some copies. There's more copies of this gene, and the expression level is higher in this gene than this gene. And we are hoping that RNA sequencing can help us solve this problem, okay? Which is great. And Microarray is doing a pretty good job on this front end as well. But yes?
Microarrays don't do a very good job of that.
Are
Hugely different, you can see that. But because they have to hybridize,
yeah.
the design of the probes affects the sequencing.
So is RNA sequencing. You have mapability issues and some other issues as well, right? So, but the point is that they are in the same market. They are solving the same level of biological problem, and which is the gene expression levels, okay? However, transcriptome is really not one-dimensional. And the second dimension will be the splicing variations. So you can see that this is one gene, and there's 3 exons here. And the splicing occurs and where the intronic regions will be spliced out. So in one biological condition, you can see that the middle exon has been kept in the final product, and in the other one, and it has been spliced out. So this one gene and eventually becomes two different isoforms, and whether we have the technology to measure that. And RNA sequencing and intrinsically can do this pretty well. There are special forms that microarray can do this as well, but you have to use exon junction arrays and something like that. It's a very special form of design, but RNA sequencing really improved the quality of the splicing isoform identification. And the third dimension for this will be the variations on the transcripts. And we know that SNP arrays can do this type of thing as well. But clearly that RNA sequencing can do this to a certain extent better because you really intrinsically look for not only the expression levels of different genes, but also the sequences you already get it. But if this is one example, just to show the idea that the reference genome in this location is a G, in your final product you see half G, half A, and you will know there's a heterozygous variation in that region. So those are something that array cannot do, but RNA sequencing can do. So the point of this slide is transcriptome is just really more than gene expression. It has multiple dimensions, and actually every single of this information will determine the phenotype. So combined together will determine the phenotype. And fortunately, the RNA sequencing can do most of these things reasonably well, not perfectly yet. Okay. The aim of the RNA sequencing experiment, the first one is to quantify the RNA abundance. So we want to know that for each gene that how many reads are there or how many copies of transcripts expressed. And the second aim is to determine the transcriptional structure of genes, including the start site, 5' and 3' ends, and splicing patterns, and things like that. So not only how much this gene is transcribed, but also how it is transcribed, where it starts, where it ends, where it has been spliced, and things like that. So determine the gene structures. And the third one is to quantify the changing expression levels of each transcript during development and understanding the different controls. So this is just using this, you can study how different genes are regulated. And the last one is to identify the variants on the transcripts. And these are all part of the aims of the RNA sequencing experiment. Okay? And this is probably the first review paper on the RNA sequencing, which was published in 2009. And this gives you the general workflow of RNA sequencing experiment. Okay. You got an RNA molecule, and this RNA is polyadenylated. So what we are going to do is you will first do the fragmentation, either fragment break the RNA directly or break the cDNA, and then you put the linkers on those RNA molecules, and then you go ahead to sequence it. So you can see that some of the reads have this poly A attached to it because it happens to be towards the end of the gene. Okay? And after you get these signals, and then you will be able to do bioinformatics analysis, the first thing to do is align these reads back into the human genome or whatever genome that you're studying. So some of the reads are in the exon regions. So you can see this is the exon, this is the exon, this is the intron. Some of the reads are in the exon regions. We call it exonic reads. And some of the reads are kind of half-half, and half in this exon, half in this exon, because the intronic region has already been spliced out. And this is called the junction reads. And the third part is called the poly-A reads. So you can see there's a start to have a little bit sequence and then goes to polyadenylation. So it really tells you exactly where the end of the gene is. Yes? We'll talk about that a little bit. Ribosome is a pretty annoying thing to But yes, we have to get rid of it. Yeah, we'll talk about that a little bit. All right. Yeah.
How do you have this poly-A and
If your reads are, I mean, we don't do that regularly because our reads are in general pretty short, and like a 36 base pair or 50 base pair. But once you go to 100 something base pair, if you have extensive chunk of a poly-A, and you can first to find those rays and where the poly-A starts, right? There's a bunch of connecting A's there towards the end of the rays. And once you find it, you remove it, but make a mark, and then align the rest of the rays. And that will help you to define the end of the gene, which is a really nice feature. We don't do that in our lab regularly on this yet, okay? Hopefully we'll do that in the future. So everybody got what he was asking, right? All right. And then after that will be the analysis part. So it sounds like pretty simple, but you know that for each individual step, both biochemistry part and the bioinformatics part can be pretty annoying. Okay, but I'll give you a trick. Okay, so as a biologist, what you do is you get a total RNA, and you send that total RNA to GNAT, and the GNAT will send the data to me. And then that's the end of it. So it's kind of pretty straightforward if that's the case. All right. So RNA sequencing seems not very difficult to do, but what we can really get out of it, we have some previous slides to show a little bit on that. And some of the students already seen this picture in other class. Okay. So from the RNA sequencing, the first thing that we can measure is the gene expression levels, right? So one gene expresses this much, the other expresses this much, or in different conditions, and for the same gene and the expression level, you can do a certain level comparison. And after that, you can identify the differential expression, and these are the things that I'm going to talk about today. And the second level is alternative splicing. So you can see that in one scenario, there's 3 exons in the final product. In the other scenario, there's only 2 exons. And if your data looks like this, you probably can guess that there are maybe 20% of this isoform plus 80% of this isoform, right? And if you are smart and devoted to look at this data. All right, things will become more complicated once the number of isoform increases. There's impossible you can do that by guessing, but there are some statistical models that can help you to solve this. But the point here is you will be able to study alternative splicing using RNA sequencing experiment. And the third level will be the transcript variation. So this is a G, and this is this homozygous SNP you can see. And for this scenario is a heterozygous SNP. The reason that we can do this, again, is straightforward because we not only see the number of rays aligned to these regions, but also their sequences as well. So if you can see consistent variation identified, there is a transcript variation. And next, we can do a lot of bioinformatics on that. So our goal is never just to identify the variation. We want to make sense of it. Okay? And one thing that we can do is if we look at the variation, if some variation is a non-synonymous SNP, and we can further look for whether they will change any structures of the proteins. Or if this is synonymous SNPs, and that will potentially affect the gene regulation, including splicing or any other type of RNA processing, like translocation and RNA editing and things like that. And if this variation locates in the 3' UTR of the gene, that potentially is the microRNA target size, and the microRNA, and that will affect the microRNA functions potentially. And keep in mind that for this experiment, we not only studied RNA binding protein, whether it can do that, but also we see the RNA binding protein binding results, the phenotype, which change alternative splicing. So if you really bring these together, you can build very interesting bioinformatics models to study this. Okay? Think about your class project if you haven't made up your mind yet. And since we study microRNA binding sites in this scenario, and we also identify the differential expression, whether we can bring this information together to make an integrated model. I think it's certainly possible. Again, these are really bioinformatics research part, not really just run through the pipelines, but that's where the fun part is, right? And another level of information when we use the transcript variation is we can study allele-specific expression. Okay, we have two alleles, one from mom, one from dad, but they don't express the same amount of RNA sometimes, right? And one allele expresses more than the other, and using arrays, there's no way we can identify that information. And using RNA sequencing, it's still pretty difficult, but at least for some highly expressed genes, and we should be able to do that. And another level of information that we can do using RNA sequencing data is for the non-coding RNA studies, right? And this is sometimes really hard to do using arrays, because every time you use arrays, and you have to know where they are and where the gene is. So most of non-coding RNAs, we don't know anything about them. So most people, Affymetrix doesn't put those on the arrays. So it's really hard to study in the global scale. But with RNA sequencing experiment, you can certainly do that. Okay? Now I'm going to use a couple of slides to briefly compare the RNA sequencing versus microarrays. Okay? None of these studies was done by me. All right, we did have experience, but these are the data from other people. Okay? So RNA sequencing is expected to revolutionize the manner in which eukaryotic transcripts are analyzed. This is out of this review paper. So again, when we talk about this, and we have already mentioned in several slides that microarray, the initial goal is trying to study the gene expression, and RNA sequencing is already beyond that. It can study the splicing and the variations. But let's just compare to study the gene expression part. Okay? And I think I already showed these slides in the first lecture, if you remember, but I will just go over this again. And this slide from Affymetrix, but actually I copied from one of Jeanette's lectures. And so this is the basic principles of Affymetrix array experiment. So you can see that this is the microarray, which looks pretty big here, but actually it's the size of your thumb. It's very, very small. And this surface is divided into many different small compartments. Okay? Some of them are over a couple million compartments. So you can see that each part is very, very tiny. And then if we zoom in this area, this is what it will look like. In one compartment, and there will be a lot of small oligos standing there, and they are all the same. Okay? And they are designed to target a certain gene, and meaning that they reverse complementary to the RNA molecules in those regions. Okay? So this is the array design. And the next step is we put those RNA molecules and can be changed to the cDNA or other form of molecules. So you can see each individual molecule here has a small red hat there, right? So which is the fluorescent or other type of labeling. And if there's a molecule that reverse hybridizes to this region perfectly, and it will stick there. But this one will not come here, but it comes somewhere else. And then after that, what you're going to do is you just detect, take a very fine image of the entire thing, and to count for how many red hats is collected in each individual compartment. Okay? The higher that particular gene is, expression level is, the more signal you will be able to detect. And that is the basic principle of it. And it has been, we have to be fair, okay? It's in the first publication of microarray comes from, I believe it's 1998, right? During the past 10 or 13 years, an array has been revolutionized a lot of functional genomic studies, right? There's no way that we can study gene expression in a genome-wide scale, and array is the first technology that enable us to do that. And there's a lot of very high-impact papers were generated, and a lot of discoveries were made. Okay, despite this, all right, there's some limitations of arrays. The first one is high background levels because of the cross-hybridization. So you are hoping that this oligo is targeting this, hybridized to this molecule, perfect match there. But sometimes, if there's only one base pair difference or things like that, you will still pick up a little signals around those regions. If this particular gene is very lowly expressed, sometimes you cannot really identify whether there is a good signal or not. So array sometimes doing really poor job for the genes that expression level is low. Okay? However, it doesn't do very good for the high signal as part as well. Okay? The reason for that is once we do the detection part of it, and you count how many red hats is in each different compartments. And if the gene expression level is really, really high, and that part will already be saturated. Okay? And let's say GAPDH, right? That is a gene that is expressed a lot and in most of the tissues. And once you see that it comes here and this is really, really bright and this is expressed a lot, but in another condition, it doubles the expression levels. Okay? But what you will be seeing here is still that big red dots because the signal has already been saturated. And this will cause the arrays has very limited dynamic range for the detections. And another key limitation of microarray is it relies on existing knowledge about the genome sequences. And because if we design some arrays to target a certain part of the genome, we already know there's a gene there. Otherwise, people don't design array based on that. And but for the RNA sequencing data, that we don't have to have that type of assumptions. Okay? For the arrays, for the RNA sequencing, there will be higher dynamic range. So we already mentioned for arrays, for the low signal end, high background noise because of cross-hybridization. For the high signal end, the signal will be saturated. Okay? But with proper depths, next generation sequencing can solve this problem really, really well. Okay? And this is the comparison between arrays and the next generation sequencing. So you can see that this X here is the signals picked out by the RNA sequencing. Okay? Y here is picked out by the microarray, same sample. Okay? In the middle range here, so it's not really too high or not really too low. So you see this reasonably well correlation, and I can read here, the correlation here is 0.51. Okay, it's still not that great, but it's pretty good. But for the lower end in this region, and the correlation here is less than 0.1. It's 0.09. For the higher part of that, and the correlation is 0.177. Okay, this really demonstrates that the dynamic range of RNA sequencing and the microarrays are different, and the array really only works well in the middle part of it. Okay, and I will when we get to the published examples, there's one striking figure that you will also be able to see, and once we get there. And the next thing is RNA sequencing shows lower false positive and comparing to the arrays. Okay, and this figure is a precision recall curve. Okay, let's go through that slowly. So what is a precision recall? Okay, before we get into that, let me tell you their experiment a little bit. What they use is a platform, microarray platform called tiling array. Okay, how many of you know this technology, tiling array? All right, a couple. So basically, it's still microarray, but what they did is they want to achieve the same goal as we are doing on the RNA sequencing. So instead of just design the probes to target on the genes that we're interested in, they target the entire genome. Okay, so you can think of they design the probes of 25 base pair, and every 35 base pair they will put a 25 base pair probe in there, regardless whether we know there's a gene there or not. Okay, of course we only target on the part that are not repetitive regions, which is half of the genome. And for the Affymetrix platform, and I think the first platform comes out in around 2005. And at that stage, they need to use 7 arrays to tell the entire human genome. And that is equivalent to, I think, 13 million different probes or 13 million features. That's a lot of information. If you do the experiment, it will cost you a lot of money as well, because 7 arrays, and most of the region we know that there's nothing there, right? We just don't know where they are, okay? So what they did is they used this platform, okay? They put the RNA molecules on the tiling arrays, and they want to see how many exons, known exons, will be detected by my technology. So X here is called a recall. Okay? What recall means is the fractions of exons being recovered by my detection. If I have 100,000 exons in the human genome, if I put samples there, I want to see, based on my technology, how many of these exons has already been expressed, been detected. Okay? So that's a recall. And the Y here is something called a precision, okay? Which means that if I detected that there's a region here, what's the percentage of detection, we know there's an exon there. So in terms, this is really more accuracy and how complete things are, okay? If you still don't get it, and go back to think of it, but the point here is you want the precision and recall both pushed to 100% as much as possible. We know it's not possible. It's impossible, right? But you still want it to be the high, both these two measurements as high as possible. But this is the performance of microarray technology, okay? For the RNA sequencing, and the data becomes much, much better. So you can see that there's multiple lines here, and each line represents a sequencing depth, okay? So you can see that this one is not as good, so because we really want it goes to that corner. And but this line, they only use 200 million base of information, which is equivalent to how many? 4 million mappable arrays, which is not too many, okay? And once we push this to 2.1 gigabase of coverage, so you can see that it really goes to that end as much as possible. So this figure really tells us that RNA sequencing has a much lower false positive. It's much more accurate in detecting the transcriptome. Okay? Are you still okay with this? Yeah. What is this? Oh, this is the analysis methods. They compare it. So they can do a certain level of normalization, whether they do RMA or we don't have to get there. But this is a different level of analysis. And this is a very hard paper to read, actually, published last year in PLOS Biology. They also have a few other papers, but this is a really, really important figure for you to study. So just get it, and how you will put that into the Encore system. So just take a look at that. No, it's more like the statistics and the biochemistry part. So there's not too much bioinformatics in that paper at all. Yeah. Right. So you are saying why it performs better in RNA sequencing, right? Yeah, analysis, but the biochemistry part, the analysis part, and both are the killing factors. And we are not talking about how much time to run on this analysis yet. Okay? And another thing is RNA sequencing doesn't rely on known gene structures, and which arrays does. And for the protein coding genes, we can study gene expression, alternative splicing, allele-specific expression. For the non-coding RNAs, this is the most important part. So you can study long non-coding RNAs or microRNAs or various forms of novel RNA species that we have never heard about. Okay? So we know that when we design arrays, we have to know there's something there to put things on the array. But for the non-coding RNAs, we know too little about these things. And so we don't have the array to target each individual applications. And another thing is, once a couple of years later, there's another type of non-coding RNA is discovered. So you don't have to redo your experiment. What you need to do is just go back to your data to look at that, and then the new biology will be in your old data. Okay, so that's really, really nice thing. And some people also are interested in repetitive elements, which is not optimal using RNA sequencing to study that. But some people discovered that in certain disease, that certain type of repeats are more than the others. Sometimes it can generate different phenotypes, which in arrays, they will never put any repetitive elements on the arrays, right? This may not be very important, but it's possible using RNA sequencing technology. And you can also study gene fusion or transsplicing, which is almost impossible to use array to study. Okay, so we will have intensive part to talk about the gene fusions in the next lecture. And this is some of our data. So you can see that this is two examples. One is breast cancer, the other one is thymoma. Actually, the next guest lecturer, Milan Rajovic, and actually he produced both of these data sets. It's a really, really nice study. So you can see for the breast cancer, there's these 10 are the normal breast cancer. These are normal breast, it's not cancer, and these are triple negative breast cancers. And these are different stage of in the month. So you can menstrual cycles. So you can see that the data really perfectly clustered together. And this one for the thymus cancer, this has become even more striking. So most of these pathology information, different subtypes of those cancers are done by Sunil, by the way, which is one of our best pathologists. And but we really don't know whether it's correct or things like that. But after we do the RNA sequencing experiment, after we cluster data together, and we have seen that Sunil did a pretty good job, right? So you can see different colors they cluster together for different pathology, different subtypes of thymus cancers, and they go together. And so these are for the human samples, and these are really, really difficult experiment to do. We know human has a lot of variations, but the fact that the RNA sequencing is so sensitive to detect these subtle differences is really, really striking. And we have some other examples to show that in clinical patients, not really in clinical, but in the real patient samples, and the data really, really cluster together, really, really tight. So those are very interesting observations to look at. Overall, for the RNA sequencing data versus microarray, so you can see that RNA sequencing has, in terms of background noise, it's much lower, and you can simultaneously map transcript regions and the gene expression, and this is yes. And dynamic range is much, much higher. For the arrays, dynamic range is a couple of hundredfold, meaning that the lowest reliable detected gene and the highest ones, they can be only a couple fold, hundredfold differences. Well, for the RNA sequencing, it's larger than 8,000-fold, and in some cases, it's over 10,000-fold. So its dynamic range is much, much higher. And ability to distinguish different isoforms, arrays is really, really limited, but for RNA sequencing, and we will get to their next lecture, and it's doing much better job. Ability to distinguish allele-specific expression, yes, we can do that. And the required amount of RNA, and what is written here is for the array is high, for the RNA sequencing is low. And but this one is a...
My experience, that's not true.
Yeah. Genetic experiences, this is not true. It also depends on the technology being used, and we are doing solid here, although I'm going to introduce one single cell example using solid technology. But if you...
The thing is, if you can do it with full amounts of RNA sequencing,
for some other sequencing platforms, like a helicose, it's intrinsically working on single molecules. So for some of the next generation sequencers, that this is true. Probably not for solid and not for Illumina as well. Right? And the last one is for the cost and mapping transcriptome to a larger genome. Here's a high, here's a relatively low. I don't know why they put it in there. But the point here is it seems for us that in most of these measurements that RNA sequencing is doing a better job than microarrays. Okay? This is one of the examples of what the data will look like. Okay? So let's go through this slowly. So you can see that this is one gene. There's so many different exons, and there are introns, right? We observed these rays, which is called exonic rays, meaning those rays only fall into the exon region. And we have seen many of these spans, okay, and which they are the so-called junction rays, which means that for this particular rate, one end in this exon, the other end is in this exon, okay? And once bring this together, we have seen a very interesting alternative splicing occurred here, which means that this rate really gives us the evidence that these two exons, they are together, and splicing occurred in this way, okay? And this rate gives us the evidence that these two are together. However, this rate gives us the evidence that these two are together where the middle one has already been spliced out, okay? And we can see that in this one single experiment, we have seen the mixture of multiple isoforms. Some rays supported the inclusion of the middle exon, others support the exclusion of the middle exon. And if it goes to the annotation of the genes, and actually there are two different isoforms, one with and one without this isoform, this exon. So this is really, so you can see the data looks like this. If you see the potential alternative splicing, it's not only by your inference based on certain level of accumulation, it is based on the data you see with your eye. And it's more trustworthy for me for that. Okay? But the question is do we still need arrays? Is FA matrix going away? Is the Illumina, the array part going away? All right. So this is just for the sake of fun, right? I got this from a website, and FA matrix CEO, they said, to achieve comparable level of sensitivity, the sequencing experience cost 10 times more than arrays. And this is what their comment is, and to achieve comparative level of sensitivity. And took much longer to complete. Even at the increased level of efforts, the array had a higher level of sensitivity at the exon level. Even deeper sequencing will be required to address low abundant transcripts as well as the array did. The array had already been implemented in multiple sample clinical programs involving thousands of samples. And as the author states, this author refers to a paper that published comparing the performance of two technology. This platform is anticipated to have a wide range of applications and high throughput clinical studies. We believe array is still alive. So that's basically what they are saying. Well, actually, it's true. In terms of in the research market, and if you study animal samples or cell lines, and probably you can use RNA sequencing because it gives you more information for the things you study. But if you want to study clinical samples, we know the variability is so high that we need a lot of different samples. Arrays can be relatively cheap on that end. And the RNA sequencing is still very expensive. And also there are, if we are, in terms of clinical application, if we already identify the markers, we don't really need to do the RNA sequencing to do this type of global profiling of everything. We can only design the best markers on the array and run through the chip. And I think the RNA sequencing development really pushed the FA matrix more towards the clinical markets, and which probably is a good thing for them. Okay? The conclusion here is array is not that. They are still have their specific application. Yes?
You also mentioned the targeted sequencing.
So Jake is asking the targeted sequencing. Actually, targeted sequencing is on the DNA sequencing. So it's not really trying to measure the gene expression level. So it's It's trying to identify the genetic variations in the exons. It's not the RNA part of it. And this part is trying to compare the RNA sequencing with microarrays. And there are certain applications that, but it's still on the research part of it, and trying to target low abundant transcripts. So people design the chips to target on these very low abundant genes, and then they kind of hybridize on that and only focus on that part to do the RNA sequencing. There are certain specific applications on that, but it's still work in the progress, and I don't think that will address the issue of RNA-seq. Okay? Are we okay still? All right, still early to have a break, so let's go through to the next topic, which can be brief as experimental considerations. Okay, once you do, as a biologist, when you do the RNA sequencing experiment, the first thing that you want to ask is how many initial materials that I need to provide, right? This is not the blood samples. This is not the DNA sequencing, which is you got the blood and you do the work, right? Although we don't have a limited amount of blood, but we still have a lot, right? But sometimes for the RNA sequencing, you have to go back to the original tissue. It can be really, really difficult, right? You study alcohol, you don't get people's brain. And if you get it, you really want to not use that very aggressively. You want to save it, right? So it's our concern that how much initial material that do we need to use. And the first study published in several Barbara Voss group, and I'm going to introduce that study in a couple of slides. And they use 100 microgram of total RNA. That's a lot of total RNA, right? But that is for technology development part, which is fine. And for most of our applications, especially in the Center for Medical Genomics here and the genetics part of it, and we are having solid sequencing, and we are working very comfortable with anything larger than 2 microgram. Am I right? Am I still right? No. Okay. They are doing pretty well with 400 nanogram. But if you talk to Howard Edinburgh, they will say, I want 5 microgram. And yeah. And but my take-home message here is I will still provide 2 microgram if you can, right? And of course, they can work on lower amount, and which is a good thing. And I have collaboration with an investigator in the University of California, and what they use is 15 nanogram of total RNA. That is really pretty impressive. And I'm going to give a few words about a paper published in Nature Methods, and they did a single cell RNA sequencing using solid platform. Okay, so when RNA is limiting, approach to amplify small quantity of RNA really exists. Yeah. It's without amplification, right? And that was really impressive. I mean, that investigator is basically a technology development type of person. All right, it's not really a biologist trying to address certain biological questions. And the second consideration is ribosome removal. Okay, I know this picture is very much like a subway map in New York City, but actually it's the structure of ribosome. Okay, and we know that ribosomes are highly abundant, and it's larger than 90% of total RNA. And some people say it's larger than 95% of total RNA. My experience is if you don't do anything about that, all your data is ribosome RNA. And we had one study in collaboration with an investigator in, I think it's in Columbus, Ohio, and a hospital there, and they study bacteria, okay? And they are trying to do RNA sequencing and using Illumina platform. And we are very happy about that, and say, good, let's do it. And then what they say, and I mentioned about this ribosome removal issue, and he said, well, since our genome is so small and it's bacterial, and we use 1 lane of Illumina, it should be plenty of it. And even though 90% of ribosome RNA, we get rid of it, and we just analyze the remaining 10%. We still can cover this genome pretty well. But it turns out that I don't know the reason for that. Over 99% of our reads are on the ribosome RNA. It's one of the reasons probably I don't know the bacterial, the proteotics system very well. The rumor is they don't have the the polyadenylation. So they don't really do the protections. So those RNAs really degraded pretty fast. All right. Once those meaningful RNA degraded, and the only thing left there is ribosome RNA. That really costs us a lot of money and a lot of trouble to analyze the data. Right? But fortunately we got it through, and we convinced them that ribosome depletion is needed even for small genome bacteria. Okay. And there's a couple of ways to do the RNA ribosome removal. And the first one is the ribosome depletion case. There's multiple of them, and ribominors and things like that, which is very good. And another way actually is to do the poly A selection, right? Because we know that that for the messenger RNAs and all of messenger RNAs, including certain level of long non-coding RNAs, they are polyadenylated. Okay. So when you do the reverse transcription, you use the poly T, and you probably can get those messenger RNAs down, but you will get rid of ribosome RNA because they are not polyadenylated. Okay, and I will talk about it in the next slide. There's issues on that as well. Or some people using enzymes that selectively degrade uncapped RNA, which is another way to do that because the ribosome RNAs, they are not capped. But anyways, ribosome removal is a big deal. You have to do it. And our experience is if you do the poly A selection, which we have a couple of experiments on that, and your ribosome information is very minimum. It's 1% to 3% of ribosome RNA. But if you do not, if you use any of the RNA depletion keys, it depends on what do you use and how good you are experimentally. And our experience is anywhere from 15% of reads are ribosome reads to 80% of reads are ribosome reads. So if you think of your data experiment, you spend a lot of money on there. You think I sequenced really, really deep. I did have 50 million reads to cover the transcriptome. It ends up only 1 million or 2 million are usable. The rest are the ribosomes. It's a pretty bad thing to go through. But anyways, these are something that you really need to be very careful on that. And the third concern, the consideration actually is related to the ribosome depletion, is to do the, is this going to be that? No, that's okay. Reverse transcription part, which means from RNA to cDNA. How are we going to do that? So the point here is our sequencers, they are second generation sequencing, they are DNA sequencers. They do not sequence RNA. Although people are working on the protocol to do the direct RNA sequencing, but it's not mature enough yet. So you have to do at certain stage to convert the RNA to the cDNA. And there's multiple ways to do that. The first one is to use the oligo-DT, which means that if this is a gene, there's an RNA molecule here. It's our hope that all the messenger RNAs has poly A tails there, okay? And then what we can do is when we do the reverse transcription, we use the poly T, and then the reverse transcription will happen, okay? And for this, the good thing is it focuses on only poly A adenylated transcripts, and the data becomes much, much cleaner. And sometimes, those are the information you need it. And the problem for that is it will generate the bias towards the 3' end of the transcripts, okay? Because we know that RNA will be degraded, okay? If it's degraded somewhere in the middle, and only the later part can be reverse transcribed, because once anywhere if the later part are degraded, and you won't get that molecule. So we will see very strong 3' bias, meaning towards the end of the gene, we observe much larger signals than towards the beginning of the gene. And sometimes that is really not a good thing to go through. Especially some of the genes, they are having alternative promoter, meaning their starting site is different. And one of your experimental procedure goals is you want to see which promoter they use. But when it goes towards the beginning of the gene, the further it's away from the poly A, and the less signal observed, then that really interferes with your conclusions, and that can be annoying. Okay? So another solution for that is instead of using oligo-DT, you can use random primer. The good thing of that is there's no 3' bias. Okay, so everything is uniform. At least it's hopefully, and can be used to study non-poly A transcripts as well. This is very, very important because we know that for the messenger RNA, for certain kind of link RNAs, they are polyadenylated. But for some other RNA species, they are not. For example, the precursor microarrays, and they are not polyadenylated. If you did using oligo-DT, and those information will be lost. Okay, and who knows? Tomorrow somebody discovered another type of non-coding RNA or coding RNA without being polyadenylated. Who knows? And then those information will not be in your data if you use oligo-DT to do the reverse transcription. Okay, and the limitation of random primer is sometimes it doesn't get rid of the ribosome RNA as well as the oligo-DT approach because ribosome RNAs, they are not polyadenylated. Okay, so there's a balance here, and our experience is now it's mature enough just to trust the ribosome depletion case and go towards the random primer part. Yeah. I'm sure they have that. Yeah. Jeanette, do you know that?
I don't.
You should find out?
I will find
She will find out. All right. All right. The next thing about sample preparation that you want to think about is whether you want to maintain the strand-specific information. Okay. So here is what we're talking about. So if this is the gene, we know this is on the DNA, but the RNA is either from the positive strand or from the negative strand. Okay. In most of the cases, not on both. Right. So it's only on one strand. But for some earlier RNA sequencing protocols, especially those based on the Illumina technologies, they cannot distinguish that. They cannot distinguish whether the sequencing from the positive strand or negative strand. And the end result for that is you don't know. I mean, you see the equal coverage of both two strands. And but however, for the novel transcripts identification, remain strands are really, really important. This is one of the figure generated by Al. So he is studying non-coding RNA. We know that for these non-coding RNAs, the annotation is really, really poor. Right? We are not even convinced whether there is a RNA molecule. There should be an RNA molecule there. So, but the hope is if there is a real signal, you will observe its intensively biased sequencing rates from one strand versus the other strand, because it's not that likely that one is this strand, the other one is this strand on the same location. It's not very likely. If you use the protocol that doesn't retain the strand information, you are not able to find that. However, if we use something that maintains the strand, you can see there's some of the molecules that biases towards signals biased towards the positive strand, others towards the negative strand. So you can increase your confidence that you are seeing the right molecules. Okay? So this for novel transcript studies, remaining strand bias is very, very important for better characterize the strand information. Even for the known genes and transcripts, sometimes people are interested in that because you can, in theory, identify the antisense transcripts and based on this type of assays, and some people are interested in that. Another consideration is whether you want to do paired ends or single ends, and this is, of course, the pair-end RNA sequencing provides more information for isoform expression. So you can see that this is for single end, and this is alternative splice region. This is intron retention, and there's some reads covered there, or sometimes there's a break out here, but once you do the strand pair end assay that you will observe much more information, and that in many cases that helps you to identify the gene structure much better. Okay, and especially this is another type of scenario is to study gene fusion. Okay, so you can see that this gene is in one chromosome, this gene another chromosome, and they shouldn't be together. But we know that in some of the cancers, and these two things, they kind of fuse together in the DNA level, and once it's transcribed, and it will become a molecule, RNA molecule has one half of this gene, another half of this gene, okay? And sometimes that recurrent pattern will cause cancer and actually metastasis as well. So if you want to identify gene fusion, you can pretty much forget about using single end because it's just too difficult and the ends are too small to do that. Although there are some studies doing that as well. And for the pair end, it will be much, much more easier. So you can see if it's one end on this part of the chromosome, the other end on this part of the chromosome, and if that information you observe a lot, and that suggests that you are observing a pair end gene fusion event. So the statistical considerations, we know that people are really greedy, right? So you spend money, you want to get the most out of it. So if you want to talk to biologists, often they will say, okay, how many number of conditions can I do? Can I do 10 conditions? And as I said, hold on a little bit, probably you do 2 conditions and do 1 experiment well. Don't try to profile everything perfectly in a single experiment. Some people want to study dynamics, dose effects, or tissue specificity, so they really do a lot of things. And the number of replicates, and for the biological replicates, and also increase the statistical powers as well. And as a statistician, that people, which I'm not, but for the statistician, they like to see more biological variance and variations included. But every single biological variation, biological replicates you included, that increase the sequencing costs dramatically. And the next is the depth of the sequencing. And do you want to use 2 million reads to cover the transcriptome, or 20 million reads, or 200 million reads, right? So those are very, very important questions, and it really depends on what you want to get out. If you want to study gene expression, and that the depth of reads doesn't have to be too much, but if you want to study a little specific expression, and you want to go much deeper, okay? And most people just want to get all these things taken care of or increase everything. But the problem is we have constraint factors here, right? And this is our constraint factor, that we have only limited funding that doesn't allow us to do a lot of things. So you really need to do certain level of balance and in the statistical design part. So you want to spend your money wisely. And I initially wanted to talk about experimental design for the RNA sequencing, and which I'm not an expert of. And the good thing is Bill Murrow from Purdue University, and he will be here for, I think, 3 lectures after this. And he will talk about an hour and a half and on the statistical considerations for the RNA sequencing and DNA sequencing experiments. So I'm sure that we will all learn a lot from him. Okay, let's take a 10 minutes break.